The overall goal of this procedure is to use the Gibson assembly approach to create large, complex DNA constructs that combine short and long segments from several different DNA sources. First, computationally design the complete nucleotide sequence of the plasmids, and then design overlapping primer sets. Next, DNA templates for the assembly reaction are amplified by PCR. Next, a purification of the PCR products is performed. The final step is the assembly reaction, transformation of the resulting plasmids, and plasmid clone verification. Ultimately, the Gibson assembly reaction is used to develop in parallel plasmids that contain flag-tagged versions of the wild-type and mutant CSTF64 proteins under the transcriptional regulation of the human elongation factor 1-alpha promoter.
The main advantage of this technique over existing methods like traditional DNA cloning is that it can create in parallel similar in design wild-type and mutant plasmids without sequential cloning steps.
Design a continuous nucleotide sequence to represent the final plasmid. Now, list the actual plasmids and DNA fragments that will be used as templates in PCR. Order DNA fragments that are not readily available, such as different combinations of tags and promoters, as single or multiple synthetic double-stranded DNA fragments.